Welcome you all to the session under the DBT STAR College scheme. We have Professor Rahul Das with us from ISER Kolkata. So to come with the formal inaugural session, I would like to request my colleague Shondipon Ray to please conduct the session, who is also the departmental coordinator of the botany for the DBT STAR College scheme. Shondipon sir, can we have you?
Can you hear me?
Yes, you are audible,
Thank
sir.
you, madam. Thank you, madam. So, welcome you all, and good evening to all the participants, resource person, or respected principal, sir, and welcome you all to this online lecture series by Dr. Rahul Das, Associate Professor, Department of Biological Sciences, ISER, Kolkata. So, today we're going to hear an exciting lecture by Dr. Rahul Das regarding the signal pathway through the plasma membrane involving receptor tyrosine kinase. So, to start with, I'll like to invite our respected principal, sir, to deliver the welcome address for this session.
Yes, sir. Shondipan, sir, actually, I got a message right now from sir.
Okay.
Sir is stuck in the BLRO office. So it will be so nice if we can go on with the session.
Directly go on to the sessions. Okay.
Yes,
So in that scenario, so I'd like to request our colleague Shumona Dotto, Dr. Shumona Dotto, to please introduce our resource person for this session.
Thank you, Shondipon sir. I am delighted to introduce our esteemed guest speaker, Dr. Rahul Das, an associate professor in the Department of Biological Sciences at ISER, Kolkata. Dr. Das has earned his PhD from McMaster University, Canada, 2008, and his MTech in Biotechnology and Biochemical Engineering from IIT Kharagpur in the year 2000. He also served as a postdoctoral fellow at the University of California, Berkeley. His primary research focus is on understanding the mechanism of signal transduction across the plasma membrane, and he has numerous publications in prestigious journals. We, we are eagerly waiting for his insightful lecture. Thank you.
Okay, I can start, right?
Yes, sir.
You can hear me?
Yes, sir.
All right. So I just tell you that this is primarily all our biology student, right? Or you have people who are non-biology as well?
Chemistry students are also there, sir.
Very good. Because I can tell you that, my background is chemistry and then I keep doing biology. So I see chemistry student and feel at home. Okay. So is to try to understand how signaling works in cell, particularly through this receptor, which is a tyrosine kinase. Cell also has other kind of receptor like GPCR that also work as a signaling module. Okay, so with two cartoons, I just tell you what is this signaling all about, right? And the cartoon that you can see is that there is this person is doing this cloud signaling or smoke signaling, and the one who is far from this person and hidden from him by this some kind of barrier, these are the mountain, actually can see the signal, can read it out, and does something, right? So here, these clouds are the input signal, and readouts are whatever this person is going to do. Okay. Now, there are, as far as these things go smoothly, nothing happens. Life goes on, but sometimes things goes wrong, right? People misread signals and then do something which is they're not supposed to do. Okay, and this is where the problem is for cell as well, right? When cell signaling goes haywire, we'll end up getting diseases that include autoimmune disorder or cancer or many other things. Okay, so let's start with how cell signaling works. And this is the two main question for the cell is how do cell transduce extracellular signals? And second one is how it prevents misinformation. So I guess most of this your student and audience know what is this. This is a cell, and people from chemistry. Someone told me that if you want to do biology from chemistry, it's all you to know that there's a nucleus inside the cell. Okay, so there's this nucleus inside the cell, and the rest of the cell is confined structure with a membrane. Okay, so this membrane structure, which is up here, this prevents transfer of any material, right? Because of the permeability of the membrane, nothing can go up here, and everything bounces back. Okay, so to transfer information from this side of the membrane to this side of the membrane, you need receptors or some protein structure which are decorated on the membrane. Okay, so in a nutshell, then membrane act as a barrier to the flow of information with the environment which is outside the membrane, which is extracellular, to the inside the membrane, which is intracellular. Now, how do cell does this? Cell uses simple strategies. They put receptors which actually receives the input signal and then translate that into an output signal here. Okay, so the input signal that cell receive can be of many forms. That can be of an hormone that fits into this pocket here and make structural changes at this part of the cell, and this creates an output signal. Let's take an real life example. For example, these are the GPCR, as I talk about, the G protein coupled receptors, where a input signal, which is in form of these boxes, binds, and they bind to this receptor. Once they bind to this receptor, what happened? This G protein, they goes and stick to this one, and it converts a GTP molecule to a GDP, right? And this is get rid of one phosphate. Okay, so whatever the charge of the phosphate is, they just get rid of that. Now, if you do that, this G protein goes and activates the second protein, and this converts an ATP molecule to a cyclic AMP. Here they get rid of 2-phosphate, right? And then this cyclic AMP goes and binds to this protein. It activates this protein, and the cellular signaling starts. Okay, so in a nutshell, the cellular signaling is a combination of biochemical reactions. We can say that, right? Now, if you think about receptors, and you can see that this receptor here doesn't have any catalytic domain or any enzyme stick to it, right? They are decoupled. So this is a receptor and this is an enzyme. So whenever a ligand comes in, they have to interact with this receptor. But in cells, there are other receptors where they look like something like this. There are 52 of them. These are called receptor tyrosine kinases. And they share a same structural property, something like there is a receptor binding domain, which is on the extracellular part of it. And there is a catalytic domain, which is intracellular of it. Okay. Now, if you look closely into one of these receptors, you can see that when there is no ligand or there is no information, this receptor generally remains in the off state. Okay, so the receptor binding domain here takes a structural state, and it is connected to a catalytic domain here, and the connection is mainly through a transmembrane domain and a juxtamembrane segment. right? They just hang on to the membrane, something like that. Now, this is generally an off state of this molecule, and there is no information. And this is what you call a steady state. Mm. There is nothing that cell needs to do. It just lives up there. Now, when there is a ligand binding or there is a ligand binding here, the binding of ligand changes the structure. And you can observe that how the structure has changed, and that turns on this pathway. And this is what you call an on state. Like this is exactly like a switch or an electrical switch, right? They remain in an off state, and you put a signal or something here, it turns into an on state. And once it turns into an on state, uh, there is this catalytic domain here. It gets activated, and it put phosphate molecule here. Okay, so the protein that does this kind of catalysis are called kinases. Okay, so what this protein does, if you activate them, they will just put phosphate residues here, which are in these dots. And once the phosphate residues are decorated on the tail here, it will recruit this adapter molecule. Okay, and once this adapter molecule is recruited, it starts to put together a signaling complex, and the cell signaling starts. Now, you must know that how a phosphate is added to a protein, and these are simple chemical reaction, and this is the kinase domain. It's the crystal structure of a kinase domain on the left, and you can see that there is this ATP molecule which is docked inside, and all it does is when you activate this kinase, the phosphate group which is here will go from the ATP to this target tyrosine residue. Okay, and the transfer of phosphate is what is how the signaling is initiated. All right, so what we call then this molecule as a switch, or these are called a molecular switch in a cell. How is it? So in off state, these molecules are inactive, and when there is a signal comes in, these molecules bind, dimerizes, and turns on, and they start phosphorylating this protein or target protein and turn into an on state. And this is how a signal functions as a switch. So how then cell regulates this off state and the on state? This is a fundamental question that most of the research that are going on. Why is it very important? Because if this on state is become unregulated and the kinase start to turn from an off state to an on state without a ligand, then you have a disease, right? Generally, we need to maintain this off state when there is no requirement for this enzyme to activate. And when there is an input signal, this off state is turned to an on state. But when this on state, when this conversion happens without a signal, this is when we have problem. And this is what we call a misinformation. Okay, so I thought that we're going to talk about this kinase, which looks like something like this. We have an extracellular domain, or this is where the ligand bind, and the intracellular domain, this is the kinase domain in blue, and this is the juxtamembrane region which connects the kinase domain. And one that is here in the transmembrane is pretty much called the transmembrane domain. And this is how a VGFR is look like. Now, these VGFR molecules are very interesting class of molecule because these regulate angiogenesis. Right? This is the development of blood vesicles in our body. They also regulate organ development, and they have also many other functions. Now, these VGFR molecules remain in an off state when there is no ligand, and they remain in a monomer state. And when there is a ligand, for example, a VGF ligand here, it binds to this receptor, it simply dimerizes, and then phosphorylates its tail residue. And this is what you call an on state, and the signaling starts. Now, under physiological state, this is exactly what is required. You have multiple receptor on the cell surface, because on the cell there is not always one receptor. You will have multiple receptor on the cell surface. And the pathway on the right is what is, we call it desirable pathway. Or this is a physiological condition. When you have a ligand, ligand goes and bind to it, they dimerizes and start a signal. Now, when the protein get activated and dimerizes without a ligand, for example, on the one at the left, this is when the clinical condition starts. Okay? And the whole research is try to understand how cell maintain the condition which is on the right versus condition which is on the left. Okay? So, next, we're going to go about and try to understand how you design an experiment to test this. Now, to design experiment to test this is to you have to understand how the readout of these signals can be measured. Now, if you think that you want to measure the signal which is on the right, the one on the physiological condition versus which is on the left, which is the clinical condition, you can simply, what you do on the x-axis, you can simply plot number of these receptors, or what you call the receptor concentration, and on the y-axis, you can plot how the receptor is phosphorylated. That is the marking of these red dots here at the tail. You can see that, right? Now, if you do that for the one for the right-hand side, that is in presence of ligand, you can see a linear curve that says that as the ligand concentration increases, the phosphorylation rate, or the number of tyrosine residues that are phosphorylated, will also increase. And one in the blue curve here, this is what is given on the situation if it's happened ligand independent activation. That is, there is no ligand and you do not see any activation, and after a certain concentration, it activates without a ligand. And this is the regime which need to be avoided, because this is where you generally find when cancer cells shows this kind of phenomena, that without a ligand, they start to activate. Okay? So now the question is how the ligand independent activation is suppressed in a cell. So if you know that how ligand independent activation is suppressed in a cell, then you know how in a cancer cell you can move this activation from this side of the curve to this side of the curve. All right. So there are two GPSs. So what we do in our lab, we actually look into, there are various VGFR receptors. These are called VGFR1 and VGFR2. And this VGFR1 has a very distinct signaling pathway compared to a VGFR2. And you can see that the main receptor that controls angiogenesis in our body is actually VGFR2. VGFR1 is turned out to be a mysterious or an enigmatic receptor, because in our normal state, this receptor is thought to function as a decoy receptor. That means it will steal the ligand from this receptor and it will do anything. But under pathological condition, suddenly this receptor start to signal. Okay, so what we wanted to know is why this receptor, which are from the same family of protein, behave differently. Because if you look into the structure of these two receptors, you can see that both of them share the same structural homology, both have an extracellular domain, both have an intracellular kinase domain, and both can bind to the same ligand, which is VGF-A, yet one has a very distinct signaling than the other. So the first thing that we did in the lab is to set up an assay system so that we can study this receptor in the lab, right? You cannot study this receptor in an animal model. So what we did is we set up an assay system and try to answer this question, why VGF1 behaves distinctly from VGFR2 and shows a very poor kinase activity or catalytic activity under normal physiological condition. So what is this assay we did? We cloned this protein, which is VGFR1 and VGFR2, in an expression vector. I hope all you know what is this expression vector is. And then you take this DNA and put into these cells. So this VGFR1 and VGFR2 in this expression vector is tagged to a protein which is called M-cherry, and this M-cherry will then fluoresce red. So if my protein is expressed in this cell, the cell will express red. And since this is a membrane protein, so all the membrane periphery of the cell will express red. Now, to determine the activity of this protein, we follow the phosphorylation of a residue at the tail, and we did that using an antibody, which can then detect with a secondary antibody, which is colored green. So the cells which will color green, we consider them that these are phosphorylated. Now, simply what we do is in the x-axis, we simply put the concentration of the red light, which actually tells the number of this red molecule that are expressed on the membrane, and the y-axis, we put the concentration of this green fluorescence, which simply tells that the extent of phosphorylation. So in real life, this is how it looks like. So these are COS7 cells that are transfected with another protein, which is EGFR. And you can see that the periphery of the cell is colored red, and you can select this red region and find the intensity. You can also select the green region and find the corresponding green intensity, and you can then plot red versus green. So that will tell us that the concentration of this protein in a cell and what is their activity. Now, in the EGFR, which is also a receptor tyrosine kinase like a VGFR, you can see that the one at the top panel is the cells, or are the cells that are not being activated with any ligand. And one at the bottom panel are the cells that are activated with a ligand. And I want to draw your attention to the cell something like this here, and cell which is this. And you can see that in absence of ligand, you can see there is no activity because I didn't stimulate it, and this is what is expected. And you can see that this cell here, even there is no ligand, this is activated. And if you see this cell here at the bottom panel, you can see that these cells, when you treat it with a ligand, can get activated, right? So for us, this situation is what we want to understand. Why cells activated at a very higher receptor density? What are these cells are? Why this cell is activated on its own? And this is what you might encounter in a cancerous cell, which are expressing high level of EGFR. Now, if you do the same experiment with VGFR, and you see the same, that is, when you have low level of expression, for example, this cell here, at the top, you don't see any activation. You can see there is no activation when there is no ligand. And these cells can be activated in presence of ligand. Now, if you take cells which are expressing very high level, they can be activated without a ligand. And this activation become ligand independent. So if you plot this, you can see that in presence of ligand, you get a linear profile when you're looking into the concentration of VGFR2 with respect to VGFR, with respect to phosphorylation of a tyrosine residue, which is up here. Now, if you do the same plot for the unligated one, where you did not activate the cells with a ligand, you can see that at a certain concentration, there is no activity, but and then slowly the activation begins based as the concentration go high. Okay, and this situation is exactly like what you have seen for the EGFR. So, what you can say that this phenomenon is common for most of the receptor tyrosine kinases. So, the first surprise that we got is actually for the VGFR1. And you can see that for VGFR1, even if you express these cells at a very high receptor density on the membrane, these receptors do not get activated in absence of ligand. And they need always a ligand stimuli to activate. So when he plotted this concentration of VGFR1 as the MCD concentration and recorded the phosphorylation of a 1 tyrosine residue in the tail, which is up here, we said that in presence of ligand, it's always a linear, which is same as all other receptor. But in absence of ligand, you do not see any activity, right? As you see for the VGFR1, VGFR2. So here we have a cell that is not affected by concentration and cannot be turned on in presence of a high receptor density at the membrane, right? And this is what a desirable situation is. So, and we want that all receptors should behave like this. So then, then you have reduced the chances of some cancer, right? For example, EGFR2 are highly expressed in lung cancer, also some breast cancer. VGFR1, VGFR2 and VGFR1 has been implicated in many other cancer and the cancer-related pain. But we see that at least in our lab experiment, we cannot activate this. So that was a surprise. And then we asked why this receptor behaving differently than the other receptors. And we also look into the rate of phosphorylation. So we study how fast these residues in the tail are phosphorylated here. And we measured the phosphorylation of this tail as a function of time. We see that the VGFR1 is transiently phosphorylated compared to VGFR2, which is sustained phosphorylation. Now, this is the first signal that tells that the, the signaling pattern of VGFR1 and VGFR2 are different, right? Because a sustained phosphorylation give a different output. A transient phosphorylation of the tail or the lifetime of the tail will actually give a different output for the cell development. And that explains why the signaling for VGFR1 and VGFR2 are actually very different. Yeah. You can see that there is a signaling for VGFR1 and VGFR2 are very different. Okay, so we could explain that, but we could not explain why these two receptors that looks exactly the same behave differently. So we started to look into the interrogate the structure of this receptor, and the way we did the structure of the receptor is to look into how ligand binding induce a dimerization and how a ligand independent dimer could be formed, and what are the structural components that prevents this ligand independent dimer formation. So the first experiment that we did in our lab is to look into the dimerization of this protein called VGFR2, and we see that the long story short is that the VGFR2 can form a dimer in presence of a ligand, and we use a dimer control, which is this protein here, where we put it, we engineer this VGFR2 to dimer spontaneously. So it looks like in response to a ligand, the VGFR2 does dimerizes, and this is how. But in absence of a ligand, it actually has a propensity to form a higher order of dimer, oligomer, or some dimers. You can see that there are some cells that are distributed here that are also forms a dimer. Okay, that tells that these molecule has an inherent propensity to dimerizes, and this is why they could be activated at a very high receptor density, even there is no ligand. We look into a mutation which is a constitutively active mutation, generally found in cancer patients, and you can see that if you mutate this residue in the extracellular domain here, this protein actually also forms a spontaneous dimer, and it can be activated linearly without a ligand, even in absence, and forms it shows a linear pattern that suggests that why dimerization is important to induce a ligand independent activity. So ligand independent activity is not a different mechanism. So it follows the same mechanism how the ligand dependent activation happens, that it induces a dimer. Now, for the VGFR1, what we find is that, I'll just go to this, is that in absence of ligand, as for the VGFR, unlike VGFR2, the VGFR1 does not dimerize, and it does not show any propensity to dimerize. So this is one reason why this molecule cannot be activated even at a very high receptor density. Now he asks why this protein does not dimerize, what prevents it from dimerization and activation. We start to probe the different structural motif. We look into the activity of this protein by removing the extracellular domain, and thought that this extracellular domain of this protein might be preventing it from dimerization. So what we did, we look into the activity of a protein which does not have an extracellular domain. Look here. So if you remove the extracellular domain, and if this extracellular domain prevents receptor dimerization, then this protein should have been activated. Now you do the same experiment with VGFR2. You can see that you remove the extracellular domain of VGFR2, which is up here, it gets dimerized. Okay? So it is suggested, or it looks like, that there is something more in VGFR1 that prevents its dimerization. So what else that can prevent dimerization of VGFR1? And we'll start to look into this transmembrane segment, which is known to induce spontaneous dimerization in lung cancer for EGFR. And then if you mutate a residue here at the middle of this domain, somewhere here, it will induce a very stable dimer. So what we did is we replaced the transmembrane domain of VGFR2 to VGFR1, and yet we could not activate these cells, these proteins. So certainly it looks like, unlike other receptor tyrosine kinases, the transmembrane induced dimerization is actually not a feasibility. Then what we did is we simply replace the juxtamembrane segment, which is this part of the molecule of VGFR2 to VGFR1. Okay, so this molecule is. So this is a chimeric molecule which carries a juxtamembrane from a VGFR2. So now you can see that if you do this, this is the wild type VGFR1, and this is the chimeric construct which carries the juxtamembrane segment of VGFR2, and start behaving as if now this protein can be activated in a concentration dependent manner. So obviously there is something here that prevents this molecule to be activated. And we focus our studies to find out what are the structural components that present here in this little part of the protein that differentiate VGFR1 from VGFR2. So what we did, we look into the structure of VGFR1 and VGFR2, and you can find what is the electrostatic potential of the structure. And electrostatic potential is simply the distribution of positive and negative charge amino acid on the surface of the protein. And what we find is that there is a difference in the electrostatic potential on the surface for VGFR1 versus VGFR2. And it looks like the VGFR1 carries a positive charge patch. You can see these blue patches here, which is not present in the VGFR2. Right? These are the neutral patches. And you can see that this is the juxtamembrane region that goes over the kinase domain, and you can see that there are these two negative charge residue. These are colored red, and they can probably form a salt bridge. And this salt bridge structure may stabilize an inactive form or the off state of the kinase. Whereas in VGFR2, you do not have this positive charge residue. So this negative charge residue cannot form a salt bridge. So the first thing that we did to test this, we ran an molecular dynamics simulation. And this is just a computational tool to see whether this interaction or this interaction, which interaction is stable. So you can try to measure the distance between this residue and this residue here. So what you find that for VGFR1, this residue is always between a 10 and 3 angstrom, which suggests that for VGFR1, this interaction is very stable. You can see that for a long period of time, the distance between these red dots and these blue dots are within these lines. Sometimes they go out, but again they come back. But if you look into these two molecules, if you look into these two molecules, here, you can see that the distance between these two are never within this 10 angstrom radius. So this is a distance cutoff that tells that beyond this distance, there cannot be any interaction. So this is the weakest electrostatic interaction. This is the intermediate strength, and this is the strong. And anything beyond that is, essentially there is no interaction. So what we speculate that the stability of this juxtamembrane segment here on this kinase domain may stabilizes this off-state structure. But then how do you test it? So testing is easy, right? So what we do is we mutate these residues here, the blue dots, to these gray dots, and then ask, can now this residue be activated, or this mutated protein can be activated ligand independently. We can see that we can see that they can be activated, this mutated protein ligand independently, but this activation is not as much as what you used to see for the chimeric construct of VGFR1, VGFR2, or for the VGFR2 itself. So we went back and looked into the structure, and we figured out that there is another interaction that is present in many receptor tyrosine kinases of this family, is a salt bridge between, or this hydrogen bond between this tyrosine residue and a glutamic acid residue at the activation site with this juxtamembrane segment. For the VGFR2, this interaction is absent. This residue here is now, rotated some 180 degree, and it comes out of this pocket. And so since this comes out of this pocket, this interaction between tyrosine and the glutamic acid residue is disrupted. So what we thought that maybe this is another additional interaction that stabilizes this inactive state or the off state of the kinase or the catalytic domain. So what we did is we went back and we mutate residues, or a deleted residue which is at the upstream. So we did not change that. So you delete 3 residue here. So if you delete 3 residue here, the phenylalanine will be pulled out of this location and will move out of this pocket, right? So this is what we thought. If this is true, then this construct, along with the mutation that we made, should activate VEGFR1 as VEGFR2. So this is what we did. We made a double mutation. We deleted some part of the juxtamembrane segment, and we also mutated the electropositive, that is the positive charge amino acid, into a neutral. And now you can see that in absence of ligand, this can be activated. And this is same as the VGFR2, right? Or the juxtamembrane construct. Okay, so this is where we think that is an additional regulation that is built up, both this electrostatic interaction and also an electrostatic interaction between here, the hydrogen bond, and electrostatic interaction that keeps this enzyme in a completely off state and there is no ligand. So if you make this mutation, that is this electrostatic mutation and the other mutation, that breaks the hydrogen bond between the tyrosine and the glutamic acid residue, we find that the transient signaling that we observe for VGFR1 is now changed to a sustained one. Okay, so that proves our point that how the structural component in a kinase could modulate the signaling output of this kinase, and why these two kinases behave so differently in spite of they share overall same structural architecture. Okay, so this is our final model, and this is where I would stop. And this is what we think is that the juxtamembrane segment, which is up here, plays a vital role for VGFR1 to remain autoinhibited in absence of ligand. And this is why the VGFR1 and VGFR2 also has a distinct signaling output. Now, this is a very unique case that we find, and but this does not answer questions on how VGFR1 is then activated in certain cancer. Okay, so we are currently working on it, and what it is looks like that it's actually not the VGFR1, which may be important in inducing a cancer-related pain or inducing certain cancer. It probably another protein, which is called phosphatase, can play a major role in inducing a stable active state of VGFR1. So this is something a bit more complicated story, which we can discuss some other time. So I'll stop here. So at the end, I would like to thank my group here, and these are the people who work. Manos, he is now in New York, doing his postdoc. Dipsutanu moved to UMass for his PhD. Shar Bikram is working right now. Somi is also moved to, I guess, in the US for a PhD. So these are the people who work in this project. We also work on several other receptors from T cells and B cells. And these are my funding. Okay. Thank you very much. I hope you get some essence of how what we do here and how the cell signaling work. Um,
Yeah,
so
thank you, sir. It was really nice to have you today. Amidst your busy schedule, I want to say again sorry to you. So we failed to reschedule the thing as you requested, but it was really a nice experience. We have our principal sir with us, who has just returned from the VLRO office. I would now like to request our principal sir.
okay, I'll stop sharing in the meantime.
Yes, sir.
Hello.
Hello.
Thank you, Raj sir. I was a little bit of busy with some official work in the inaugural session. I was not present there. So my sincere gratitude to you and regards, obviously. I missed the session anyway. But my warm welcome to you, in our college. I think yesterday one session was there with Professor Shubhajit Bandyopadhyay, isn't it, ma'am?
Yes, sir.
So he also agreed to come with you in your college in physical mode.
Yeah, we discussed about that. So